Hi, so my name's Courtney Hudson, and today I'm going to be discussing CRISPR-Cas9 gene editing via non-homologous end joining. Um, so just to introduce, the first part is the CRISPR-Cas9, which is commonly used to cut out very specific and large segments of DNA. For example, like segments containing mutants or are damaged, and need fixing. And the CRISPR-Cas9 cuts these off and creates double strand breaks or DBS or DSB in order to delete them. This process is commonly used in eukaryotes, but is sometimes used in prokaryotes. And then the other part is the non-homologous end joining, and without the help of the process of the non-homologous end joining, or NHEJ, many unwanted mutations, usually frame shift, occur if CRISPR-Cas9 is just left to attach it by itself without this process. The non-homologous end joining process utilizes enzymes that capture both ends of the broken DNA molecule. It brings them back together in a synaptic DNA protein complex. And finally, the DNA break is repaired. And down here shows the Cas9 entangled in a DNA strand. To dive deeper into what we just discussed, CRISPR-Cas9 works by identifying and cleaving off a large section of selected DNA and breaking it into the two strands. Once this happens, the Cas9 protein, or not once this happens, sorry. The Cas9 protein needs single guided RNA to attach to. And the Cas9 is what helps break it, but to do that, it needs to recognize PAM, or a PAM sequence, and then it makes the double-stranded break 3 pairs up from the PAM. And the PAM sequence is a short, like 2 to 5 base pair sequence or conserved DNA sequence that helps Cas9 to know where to bind to so it can break. Once a breakage has occurred, this is when the non-homologous end joining comes in, where resection, it's called. When it breaks, resection takes place where the nucleotides are lost here. As you can see, they're going away. And then they produce, since these are away, they produce these overhangs. These overhangs are useful because Ku70/80 heterodimers recognize them, and then they attach to the overhangs, which, and the purpose of Ku is basically for the DNA PKCs, or catalytic subunits, to attach. And then once those are attached, the Artemis proteins can then attach to the DNA PKCs, which causes phosphorylation, and this trims the overhangs. And then after the overhangs are cut and it's ready to be put back together, ATP dependent ligase 4 then comes and reconnects these strands back together. So just an overview of this process, just because I want to make sure it's in your guys' heads. So this is the Cas9 protein right here, and as you can see, it takes the DNA and it breaks it. And then once this is broken, again, the Ku70 and 80 heterodimers, these rings here, attach so that the DNA PKCs can attach, causing phosphorylation with the Artemis proteins, which are not shown here, but would be like under here. And then DNA ligase attaches over all of those and then uses ATP to activate and bring these back together and repair it to a non-mutagenic strand of DNA. So, what are some benefits to using the non-homologous process? Well, it's for 30 minutes to be exact due to its simplicity. While non-homologous recombination takes up to 7 hours. Or sorry, just homologous recombination takes up to 7 hours. It is simple, efficient, and relatively cheap process. The non-homologous end joining is active during the entire cell, which just basically allows a higher capacity for repair because it's always repairing, or it can always repair. And then CRISPR-Cas9, a big benefit to it is that it's easy to program, making it possible for the DNA strands to break wherever you want, due to the high sensitivity, making it very selective. And then some real world applications. So a big one that they're using the CRISPR-Cas9 non-homologous end joining for is cancer treatments. By carefully deleting the cancer-causing mutations in the cell in hopes that if these cells replicate, they won't keep replicating the mutagen and therefore won't be cancerous anymore. They're finding that this is just hard because it's hard to identify cancer cells versus normal cells. So there's still more research to be done, but it is an interesting way that they're finding to use it. It can also help modify hereditary genes, so those passed down in generations, by cutting out the harmful part of the genes. And the hope to this is kind of like, I don't know, perfecting the DNA that people have, so that these diseases don't affect them, which is kind of controversial, but anyhow. As well, specifically, it was shown to help improve vaccines used for tuberculosis by using the CRISPR-Cas9 non-homologous end joining to purposely cause mutations in tuberculosis or delete them if they're helpful in the hopes that you render the tuberculosis ineffective and inactive. And again, it can be used to help delete these mutations, as you can see here, like frame shifts, which again can cause cancer or just other problems. And then here are my references. Thanks for watching.